Hey, good morning, good afternoon, good evening, depending upon where you are right now. Welcome everyone to virtually the One Health Microbiome Center and happy World Microbiome Day. It's fantastic to see you all spending a small sliver of your day with us at a moment of, I think, profound transformation in the life sciences. I'm Seth Bordenstein, director of the One Health Microbiome Center here at Penn State, professor of biology and entomology. And it's a pleasure to welcome you to a very special seminar and opportunity to host critical conversations and do that with visionaries around the most important topics in our field. The pace of technology driving the microbiome revolution is breathtaking, and the science itself arguably remains increasingly complex. And so at this moment, we need hubs of microbiome communities to lean in and to convene together for knowledge and collaboration that shapes our future. And that is what today is about. That is World Microbiome Day's mission and contribution. And our speaker today is at the forefront of both these topics now and for the future. And so we are thrilled to welcome Dr. Curtis Huttenhower, a leading authority and pioneer in human microbiomes and public health. He needs little introduction to many of you, I'm sure. Dr. Huttenhower is a professor in the departments of biostatistics and immunology and infectious diseases at the Harvard T.H. Chan School of Public Health. He's also the co-director of the Harvard Chan Microbiome and Public Health Center. He's played pivotal roles in projects really that defined our field, including the NIH Human Microbiome Project, serving as a lead investigator for both phase 1 and phase 2 of this landmark initiative. And in similar vein and fashion, he now co-leads the Human Virome Program Coordinating Center that arguably will do the same to the field that the Human Microbiome Project did. So simply put, Dr. Huttenhower's foundational computational tools and research have truly shaped how we study microbial communities, including in microbiome profiling, metagenomics, and microbiome-associated disease signatures. And so this is just scratching the surface of his wide and vast scholarship at the interface of microbiomes and public health. We are grateful to have his time today. We're grateful that you joined us. We're excited to hear about the current waves of his research and how it will continue to shape us and our science going forward. Please virtually join me in giving a warm welcome to Dr. Curtis Huttenhower. Thank you so much for being here with us. The title of his talk is Microbiome Alphabet Soup, the HVP, the OHMR, and the HCMPH. Welcome, Dr. Huttenhower.
Fantastic. Thanks so much for the very kind introduction, Seth. While we're getting the screen share turned on here, I was telling Seth earlier that at what I believe was the first World Microbiome Day during one of the International Human Microbiome Congress meetings in Ireland. They kicked everything off with balloons and cake. So I hope everyone today gets some balloons and cake along with the microbiome science. If my screen share looks good, I can dive in.
Looks perfect.
Fantastic. Alrighty. So as Seth introduced, everyone likes our acronyms in science. So I'll talk about a few of the acronyms we've been working on today. One of which Seth introduced already being the Human Virome Program. So the HVP kicked off fairly recently from the NIH Common Fund as a follow-up to the earlier phases of the Human Microbiome Project, looking to really understand the baseline endogenous human virome in the same way that we set out to understand the normal human microbiome during the HMP. Like Seth mentioned, I'm delighted to be able to work with the program via the coordinating center jointly with Owen White at the University of Maryland, so that our responsibility is data sharing, data deposition, tool availability, documentation, outreach, and we get to do the fun stuff like run the meetings as well. Most of the cool science, though, comes from a series of 15 projects associated with the program. There are 5 large virome characterization centers that are tasked with assaying large populations across the body for virome data generation. And then there are a set of 10 individual studies, 5 of which are based around either computational or wet lab tools and protocols, or 5 of which are based on characterization of virome function in a variety of different conditions. So really, we expect to make lots of data in the consortium, look at new methods, new biology from the targeted studies, and then our goal at the coordinating center is to help share all of this new information. So as the NIH helps move this forward, we're just getting the consortium off the ground. And one of my lab's interests is in computational methods development. So we've been looking at ways to improve viral profiling from metagenomes and metatranscriptomes. This has been challenging historically, in as much as 5 or 10 years ago, we couldn't grow most microbes or most bacteria. We still can't grow most viruses. And enrichment protocols such as virus-like particle enrichment can be highly variable. They can be challenging to execute on certain types of samples, including human stool samples. And the same protocol can work great in certain samples and not so well in others. So it makes it hard to pull out things like viral metagenome assembled genomes or MAGs from many samples. Despite this, there have been several really good large-scale projects recently that group together all of the available metagenomes from various environments and pull what looks like viral MAGs out of them. This still misses at least half of the community. Just on average, for viruses and bacteria, about half of any given microbial community will assemble into non-trivially large contigs. Only some of these in typical communities will be viral. Only some of these will be sufficiently high quality mags to carry forward. It's hard to tell for viruses which those are. So a lot of this has added up to making it fairly difficult to have both sensitive and specific methods to find all of the viruses in an enriched or unenriched DNA or RNA metatranscriptomically assayed community. So we've been working on improving profiling for this with a system called Baklava. I should have included that in the title. Yet another complicated acronym. Baklava builds on some of the technology that we've used previously for bacterial profiling and functional profiling in unassembled shotgun metagenomes, or in this case, metatranscriptomes. There are sort of three arms to baklava, one of which looks for nucleotide matches to known viruses. And it does this by building specifically viral nucleotide targeted markers using a methodology similar to what our Metaflan software does for bacterial profiling. The second arm looks for more distant matches to amino acids using translated search of short reads, much in the same way that our Human tool does for functional profiling in microbial communities. And this is important since viruses do evolve very rapidly, so just matching known viruses using highly homologous nucleotides misses a lot of stuff. The third arm is made to reach even a little bit further out, where if assemblies and assembled contigs are available, Baklava can use a deep learning method developed by one of the collaborators, Philip Munch. And I should say that Baklava overall is a project from Jordan, who's a PhD student, and Eric, who's a scientist with the lab. The third arm will then use this deep learner to classify contigs directly as viral or non-viral, and if viral, into roughly genus level taxonomy. So at the end, we're able to integrate these three different types of very specific, broader, and machine learning based viral profiles to get an integrated view of each individual metagenome or metatranscriptome of interest. So Baklava builds on first a set of databases that are extracted from both our own assemblies in collaboration with Nicolas Agata's lab and these variety of large viral MAG databases that have been released recently. So we integrate many of these large metagenome-based databases with a couple of gold standard references from RefSeq and ICTV to be able to have both very high quality, well-characterized viral genomes, as well as these kind of noisier but much higher coverage wild viral assemblies. We integrate these using a nucleotide clustering approach that we'll refer to as viral genome bins. So this clusters together genomes that are sufficiently similar by the ICTV criteria to be considered the same viral species. In addition to this, since viruses can rearrange fairly quickly, we do a second step of clustering that accounts for rearrangement. So it will find non-contiguous homology between what is probably roughly the same virus. And these steps are similar to what Nicola's group developed for species level genome binning based on clustering of mostly bacterial mags from metagenome assemblies. Baklava takes this integrated database, assigns taxonomies to any VGB for which it's possible, either directly from ICTV or RefSeq, or using ancestral state reconstruction for less well-characterized VGBs. And then we create nucleotide markers in a way that I'll show in the next slide or two. And we call ORFs and cluster those directly into pan-genomes for the amino acid matching that I mentioned. As a result, most of the VGBs we construct are directly clustered. About 10% of them have enough rearrangement in them to require what Jordan calls superclustering, so this ability to find rearrangements of what is otherwise the same viral genome. And then over here gives a quick overview of what taxonomy we find as a result. So if you're familiar with how Metaflan looks for bacterial profiling in microbial communities, it'll identify whole genes that are unique and conserved within a clade of interest. Baklava does something like that, but since viruses have such varied open reading frames, instead of looking for whole genes, it scans for 100 nucleotide windows that are unique to a VGB. So contained by most or all of the the genomes in that VGB and not present in other VGBs. Given sufficiently many of these adjacent, we'll collapse those into a unique marker sequence, and that gives us the ability to find typically dozens to hundreds of markers for larger viral genomes, much in the same way that Metaflan does for bacteria. So given this combination of nucleotide markers that are guaranteed to be unique, protein markers from the pangenome that aren't necessarily unique but are more sensitive, and deep learning-based classification, we can then synthesize those into an integrated profile for any given metagenome or metatranscriptome. We've evaluated this using a variety of approaches. I'll show some results here using synthetic data that consists of known viruses that are directly homologous to some database element, decoy viruses that are almost homologous. They are sufficiently similar to be recognizable, but also sufficiently dissimilar to be guaranteed not to fall into an existing VGB. And then we've included novel viruses that are synthetic sequences guaranteed to be unlike anything in the database, so they should not be classified as viruses. You can think of these as spanning a range of percent identity and coverage. So known viruses have sufficiently high identity and coverage to fall within the ICTV criteria. Decoy viruses don't, but they're still pretty similar, and novel viruses are just plain different. Jordan generated a variety of both staggered and even synthetic communities, drawing from different combination of known decoy and novel viruses, with the statistics shown down here. And the important part is that we've, like with Metaflan, we've been very careful to tune for high precision and pretty good recall. For known viruses, we get great precision and recall. For the decoy viruses, where we should identify these as viral without necessarily knowing which VGB they are, both our precision and our recall drop a little bit. And then for unknown viruses, or unknown sequences, I should have just blanked this out. Our recall should be 0. That's good. Or excuse me, I should have blanked this out. Our precision is essentially undefined and our recall should be 0, so that's good. Similarly, so this is for the even communities. Similarly, for staggered communities, we have essentially perfect precision and recall on recognizable viruses, pretty good precision and recall on novel viruses, and essentially 0 recall, which is what we want on decoy sequences. So given that Baklava provides a lot more comprehensive, and I haven't shown the comparison to other methods, but as a result of this sensitivity, Baklava provides much more comprehensive coverage of viral profiling in unenriched normal metagenomes and metatranscriptomes. Given this, we've been able to do some pilot studies of the virome in conditions like inflammatory bowel disease. And this draws from work that we did during the second phase of the Human Microbiome Project, where to very briefly recap some of the data in the Inflammatory Bowel Disease Multiomics Database, more acronyms, our HMP2 project, the IBD-MDB, followed just over 100 individuals with either Crohn's disease ulcerative colitis, or control individuals for about 1 year each. So these were drawn from a range of ages, from pediatric to adult. Individuals were sampled using a whole bunch of different methods, focused mainly on stool every 2 weeks, but accompanied by blood draws periodically, some baseline data, including biopsies. As part of the project overall, we have host-targeted information, RNA-seq, genetics, serology, etc. What we'll look at here are the paired metagenomes and metatranscriptomes from a subset of these 130 times 1 year long time courses. So this gives us DNA and RNA viruses per sample. So one of the things that we were able to see using baklava's profiling is that unlike some of the previous publications on the IBD virome using just assembled contigs, we do see viral community diversity drop during both ulcerative colitis and Crohn's disease. And this parallels what we'd expect from bacterial communities. Some of the earlier publications had indicated that viral diversity might increase, and that's true for certain clades, but only the ones that are well detected by assembly. The virome overall much more closely follows the bacterial community, as we'll see later, in large part because it's mostly phage. And this is expected in the gut. The vast majority of viruses we see are bacteriophage, and they pretty closely track their bacterial hosts. Another interesting thing was that this was detectable for disease diagnosis even during periods of quiescence. So the inflammatory bowel diseases are remitting relapsing diseases where there will be flares, periods of activity, followed by periods of remission. Most microbiome changes are only detectable during flares. For most individuals, there are relatively few differences during remission. That was only true to a lesser degree for the virome. So we could actually see these changes in virome structure even outside of disease activity. We could also see them within disease activity as well. So if we refer to active disease as dysbiosis, we again see the same drop in virome diversity. Also paralleling what happens in the rest of the microbiome in the bacterial community, we see less stability in individuals with IBD and especially active IBD. So if we look at the overall amount of variability in the population microbiome, we get a baseline from pairwise comparisons of all individuals here. So what you're looking at on this scale is how different any two people are in terms of their virome composition. Those individuals outside of active disease, so either non-IBD controls or quiescent samples from IBD patients during quiescence, tended to have a little less variability here. Those during periods of disease activity tended to have a little more variability. So again, paralleling the bacterial community, the virome is more variable during periods of inflammation. Despite this variability, viromes are even more personalized than the rest of the gut microbiome. So on average, any two individuals from a highly similar population might share something like 10 to 15% of their bacterial species. The virome is even more individualized. So if we look at the amount of within subject variability, you tend to look more like yourself over time. That is less true if you're an IBD patient going through periods of inflammation, but you still look a lot more like yourself than you do like anyone else, even more so during periods of quiescence than during periods of inflammation. So your virome is even more variable than the rest of your gut microbiome. It tends to be relatively stable within individuals over time. You look a lot more like yourself than anyone else, but perturbations like GI inflammation can destabilize that even more, much as with the rest of the bacterial community. But interestingly, the composition of these personalized viromes varies tremendously from person to person. So there's multiple different types of personalization going on with viral communities in the gut specifically. What you're looking at here is one column per sample, colored by how prevalent the viruses in that sample were. So if an individual over here has mostly very dark blue, that means that most of their viruses were seen in most people. If an individual over here has mostly very light gray, that means that most of their viruses weren't seen in anyone else. So despite the fact that you tend to have your own virome over time, your own virome might be really different from everyone else's, or it might have viruses that appear elsewhere, even if they don't appear in the same combination elsewhere in the population. So there are lots of ways for your virome to be unique. And again, I would hypothesize that this is in part a function of viruses just plain evolving quickly so that it's easy for individual viruses to become personalized. Their ecology can also evolve very quickly based on things like phage-host interactions. And so some of this ends up associating portions of the virome with IBD. And I can start by introducing the process by which we look for differential abundance with respect to a phenotype of interest like IBD. We've recently updated our statistical package for this, Maslin, to a new version. I won't have time to talk about it today, but version 3 is better than version 2. We've changed the core statistical model so that it can handle mixed presence-absence logistic detections and abundance-based linear detections. There's a new median normalization for compositions that increases our sensitivity quite a bit. We can handle absolute quantification if you have available a PCR or a spike-in protocol. And then there are a few more changes to additional model designs that the system understands now. But the upshot is that there's also new visualization. So the upshot is that we can identify bacteria from normal microbiome profiling that are either more likely to be, excuse me, present or absent during disease, or more likely to be more or less abundant during disease. So something like the usual suspects of Faecalibacterium prausnitzii tend to be both less abundant and less prevalent, less detected, mostly during Crohn's disease and to a lesser degree during ulcerative colitis, exactly as been seen in previous studies. We can now apply this same model that accounts for longitudinal data, read depth, disease, diagnosis, and activity, and a couple other covariates like recent antibiotic usage, age. We can apply this same model to our virome profiling. You can see if we snip a few of the associated viruses off the top, most of what we see are either phage or unclassified, and this is actually more classified viruses than we usually see. Even most gut microbiomes have mostly unclassified viruses. There's way less taxonomic and phylogenetic coverage of most viromes than there is for bacteria, even in the well-studied human gut. So there's part of why the HVP is so exciting is that there is a lot of work to do out there. Of the viruses that we can identify, many of them are crassphage, so these are typically Bacteroides phage, or others like Foxway virus or Totatus virus that are typically either E. coli or Faecalibacterium phage. And like bacteria, viruses are generally lost and or less abundant. So like I mentioned during IBD. So like I mentioned earlier, most of these phage mostly tend to track pretty closely with their bacterial host abundances. And I hinted at this earlier. We've fairly recently gotten RNA viral profiling from the accompanying metatranscriptomes. The last time I gave this talk, it was really hot off the press. I just had the data. Now it's been a little while, but I still don't have a good slide on it. So stay tuned for both the similar statistical analyses on the RNA viruses. Plus, we're starting to dig into what some of these mostly Bacteroides phage might be doing in IBD. So I'll transition there, having talked about the HVP and baklava for viral profiling as applied to IBD data from the Human Microbiome Project, to a completely different collaboration that I thought would be fun for this audience, the OHMR, or One Health Microbiome Resource. And preview, thanks to Seth and the center there for being very helpful with the naming. We got our name around the same time that the center there did, and Seth was very kind in seeing it as a positive rather than a collision. Where if it's an opportunity to work more with the Penn State Center, I think that's only good for both of us. Where we got the name was through a really unexpected collaboration with Hill's Pet Nutrition. So as someone who works at a school of public health, I spend most of my time thinking about the microbiome in large human populations. I had the fortune to meet one of the Hills scientists, Dayakar Badri, at a conference several years ago now. and came to realize what really amazing population health and especially nutrition data are available from companion animals. So most people don't realize Hills has a phenomenal research center that's about 1,000 dogs and several hundred cats. Their whole goal in life is to hang out, play on the puppy park, and eat different foods and see what they like. There are on-site medical facilities. There are many on-site technicians that know the animals individually. It's a phenomenal facility. And these animals are tracked for their whole life in terms of their growth history, their medical history, and their dietary history. So we know more or less to the gram what food each individual animal ate every day. So this has given us a lot of opportunities for interesting studies there that we couldn't possibly do in humans. We've started out by making an integrated resource of companion animal microbiomes available, hence the One Health microbiome resource. And what the OHMR aims to do is consolidate both public shotgun metagenomes from cat and dog gut microbiomes, as well as about 2,000 or so shotgun metagenomes that Hills has developed in-house. So these are all available online now in a uniformly processed manner. We've updated many of our tools to better profile companion animal, canine and feline gut microbiomes as well. And just for comparison, there were about 500 or so published cat and dog shotgun metagenomes when we started, which have now been combined with the 2,000 additional internal Hills metagenomes, with more coming out continuously. So we're really hoping to make the resource a platform for broader and more specific companion animal microbiome studies in general. One of the first things we did given the resource was start from a very high level and say at a population level, since we've worked with many human gut microbiome populations, how do these cats and dogs compare to what we would find in humans? And there have been some studies like this before that help to identify the overall variability between people in blue, and I'll keep these colors for the next few slides. So all of the blue samples here are either from the HMP1, which was a healthy population. The darker blue samples are from an international population that was drawn as an outgroup that's very unlike the North American population in the HMP1. We tend to have a few more canine samples in yellow down here, and slightly fewer cat samples in red up here. So like I mentioned, there have been several studies now that do comparisons like this. And yes, of course, at the species level, there are large differences between what we tend to encounter in humans, dogs, and cats. We can start drilling into this, and with the more precise shotgun metagenome profiling that we have now, it's possible to find both known SGBs. So now we're back in bacterial species level genome bins. Known SGBs are those that contain a cultured isolate representative. So you can find known SGBs like, interestingly, Prevotella capri that show up at pretty high abundance in all three hosts, and plenty of unknown SGBs, or those that do not yet have a cultured isolate as part of the species level genome bin. So this gives a wide range of less well-studied organisms that are important enough in the mammalian gut microbiome to show up across these really diverse hosts. Many of these are host specific though, either to cats and dogs jointly or to a single one of the hosts. So we can pick out typically less common organisms, but those that are really highly abundant on either humans, dogs, or cats, and not found in the other host organisms, as well as those that seem to be common to companion animal lineages, but not to humans. So again, this is similar to what some previous studies have done, but really extends the level of detail so that we can see at the species level genome bin what the different ecologies might be between people, cats, and dogs. Where we got really interested in it, though, is kind of digging one level deeper into the strain genetics of these shared species, especially. So just because we all have Prevotella capri, for example, does that mean we have the same Prevotella capri in people versus cats versus dogs? Since when we've done similar studies for humans, certain bacteria will trace with human genetic lineages and thus differentiate by population. Others don't. There's more transmission, there's more gene flow, and so the evolutionary strategy of that bacterial lineage is a little bit different. So we wanted to look at that quantitatively for the companion animal data, for which postdoc in the lab, Andrew Ghazi, developed a new package for strain level epidemiology in microbial communities called ANPAN, which is not an acronym. He just made the name up, although it is another bread type, I should add. So what ANPAN does is offer three different packages for different types of strain level statistics in microbial communities. It operates from taxonomic and functional profiles, so it picks up where some of our earlier profiles leave off. First, detecting which samples have enough data, have enough sequencing depth, essentially, to support strain analysis for which taxa. Because you can only do strain level analyses for a particular species if you have enough sequencing depth to really see variance within that species. So it's easier to detect a species in a sample than it is to get its strain genetics. So OnPond does this by essentially looking for pan-genome saturation. Since we don't know the size of any particular wild microbe's expected genome, it's probably around what its corresponding isolate would be, but we don't know exactly. OnPond will ask across a set of samples for each bug, which samples look like they have saturated that bug's whole genome. So it'll cluster those samples as sufficient apart from everything else containing that bug as insufficient. Given the sufficiently covered sample bug combinations, OnPond has a model for associating strain-specific gene carriage with a phenotype of interest. So essentially finding genes that are present or absent in strains of the same species in correspondence with some phenotype or covariate. It can do the same thing with strain phylogeny, so ignoring gene carriage and instead saying, are there overall evolutionary patterns within a species that covary non-randomly with a phenotype of interest? And then finally, it has a third module that does the same thing for gain or loss of whole pathways. So is there an operon or a combination of functionally related genes that tend to be differentially carried by strains of a species in correspondence with a phenotype? So we'll focus mostly on this middle one here in comparing the lineages of shared microbes that we see in both people and cats and dogs to determine if they're really the same or not. Are they transmitted or are they independent lineages? So if we ask how many microbes are truly shared, how many strains seem to be transmitted between people, cats, and dogs, sometimes we see lots of this. So for what used to be Bacteroides vulgaris, Phocacola vulgaris, the human in blue, feline in red, and canine in yellow lineages are highly intercalated. And this suggests that there are frequent historical transmission events of Phocacola vulgaris between human cats and dogs. So we are carrying more or less the same lineages as most of our pets. Biology being biology, though, we can also find exactly the opposite. So in this example of Blauschia, there's essentially no historical transmission events. There are completely differentiated lineages between humans, cats, and dogs. So this is a bug that seems to co-evolve directly with its host, with very little transmission from mammalian host to host. And we can find examples in between as well. So for something like Ruminococcus navis, interestingly, in humans, this is an inflammation-associated bug. You can tell there's relatively little blue. It tends to be pretty rare in healthy subjects. And we can see that there's one dominant human lineage, but a lot of these individual transmission events uniformly from dogs, where it's quite cosmopolitan and doesn't seem to associate with inflammation at all. So there's both different host biology and different evolutionary biology going on here. Especially if you squint really closely, there are one or two little red tips around here. So the feline lineage is mostly monophyletic with very rare transmission events. So if you think about something like a zoonotic disease, we know that transmission of most pathogens between most hosts is rare because we notice when it happens. This type of transmission of commensal organisms for something like B. vulgaris is probably happening all the time without even being noticed. Certain bugs have a more pathogen-like ecology while not being pathogens, and we just haven't been able to see this kind of silent transmission before because there's no obvious phenotype the way there is for a zoonotic pathogen. So I think this is really helpful to keep in mind from a One Health perspective, of how much back and forth we have with our microbial surroundings, even our companion animals. I don't think my cats have wandered into frame yet, all the time without even necessarily noticing. So like I mentioned earlier, this is just our sort of first high-level study with hills and the OHMR. We have other diet-focused studies ongoing, looking at different amounts and sources of fiber, looking at not quite ketogenic, but low carbohydrate diets and how they affect the microbiome. Looking at early life development, something that's been really interesting is that as soon as a set of puppies come into the facility at Hills, all of their co-housing information is stored for the rest of their lives. So we know both developmental trajectories and contact histories for the dogs, especially at the facility, in a way that we couldn't possibly for humans. We have a study ongoing in improving the response to antibiotics. So can you change diet in a way that helps to aid the response or the recovery from antibiotics? Since occasionally when a dog is, banged up a little bit during play on the playground, they rarely get antibiotics for clinical reasons. So we're then able to see how they recover from that afterwards. And finally, some more basic biology studies of how biochemistry in the gut differs between humans, cats, and dogs in this case. So similarly, keep an eye out for additional work there, and we'd love for people to use the OHMR resource. I really am looking forward to more opportunities to work with the Penn Center, since sadly we don't get to do this very much in Boston. This is sort of a one-off treat for me, and it's been a really productive collaboration so far. So I'm looking forward to hearing more about similar work going on at Penn. I'll wrap up here at the end with a little bit of information about our own microbiome center. It's a mouthful. Seth, you nailed it at the beginning, the Harvard Chan Microbiome and Public Health Center, or HCMPH. We, of course, being located at a school of public health, tend to focus on human populations, and we do provide an umbrella for a variety of different microbiome-related activity there. And I should mention I co-direct the center along with my close collaborator, Wendy Garrett. The center has a number of facilities, resources available to help support microbiome research. We focus on research projects, both these kind of large consortium projects and a range of individual microbiome studies in Boston and elsewhere. We offer some facilities to help make microbiome research easier, whether it's biobanking. We have a specialized bioinformatics core. We have a specialized collection core that has built kits for both human and companion animal microbiome sampling. And Wendy runs a notobiotic facility for germ-free and notobiotic mice. Over the past several years, we've developed a series of courses from undergrad to grad level on the microbiome and microbial communities. And we offer a variety of year-round short courses, mostly focusing on bioinformatics. We had our first local week-long course this past summer, I guess this current summer. And we're planning on the same thing again next year, so keep an eye out for that as well. We have community events similar to this, but not quite as exciting, where we have a seminar series during the semester, an annual symposium, and collaborations throughout Boston, nationally and internationally. Especially in the past few months, we've been focusing on things like industry partnerships. So if you're out there from industry and you'd like to work with large human populations, let me know. Something that I do think is pretty unique about the center is the collection core especially. So many microbiome centers have sequencing data generation facilities, analysis facilities. Because we sort of uniquely work with these 10,000, 20,000 person large human populations, we developed a kit that is room temperature compatible, USPS compatible, so it's very cheap to ship through normal mail and customizable to collect different types of stool, oral, other body site microbiome sample types in a couple of different buffers that are all room temperature stable but compatible with different multiomics. So metagenomics, metatranscriptomics, metabolomics. We have custom instructions, both for humans and cats and dogs. We've translated them into a couple of different languages. We have some standardized mini questionnaire instruments that will get sort of very basic health and dietary metadata along with these. And to date, I think we've shipped around 40 or 50,000 of these over a variety of different studies. Some of them are huge and some of them might be just 20 people. So there's really a wide range there. And then the collection core integrates with our analysis core, which again offers kind of a specialized bioinformatics service that also integrates with things like the biobank and our data generation facilities at the Broad Institute to automate a lot of the initial sequence data handling, basic reporting, and then go on and do more custom things if needed. So I'll wrap up there. I think there's hopefully still some time for questions. I want to thank again our collaborators at Hills, Owen and his team at the University of Maryland, and everyone throughout the HVP, Nicholas Agata, with whose lab we work on many of these studies, and again, Wendy and everyone in the center in Boston. So thanks again so much, Seth, everyone, for hosting. And if there's time for questions, I'm happy to keep going there. Thank you so much.
Absolutely. Thank you very much, Dr. Huttenhower. This was clearly a small sliver of the real positive impacts you've had on the microbiome field, and I know we all want to thank you for that. Um, the research project manager for the One Health Microbiome Center is Grace, and she's working to set all this up and is behind the scenes. And I think she'll be moderating some of the questions and answers. We do have four of them prompted already that Grace can articulate and have a discussion with you. And if anybody else wants to chime in, and I'm sure there are more questions, just type them into the box and we'll spend the next 20 minutes if we'd like to get through them. So, Grace, I'll hand it over to you.
Yeah, thank you so much. And thank you for that great talk. Now I'm wondering how many microbes I share with my cat. Um, so the first question kind of relates to an earlier portion of your talk from Patrick. Is there a significant gap in library prep methods for assembling viral metagenomes with respect to things like secondary structures and RNA viruses? Essentially, how much of the problem space in characterizing viruses is computational and analytical versus sample preparation and enrichment methods?
So a really nice question. I'd say a little bit of everything, all of the above. For DNA viruses, there are great enrichment protocols that can work really well sometimes. Things get a little weird for small single-stranded RNA, excuse me, small single-stranded DNA viruses for which not all library preps will retain them. And then for RNA viruses, it's even a little bit more of the Wild West. I haven't even seen any studies of things like, say, stability of single-stranded RNA viral genomes after extraction during reverse transcription and prep. The problem then being that on the computational side, even though we've sunk a bunch of effort into baklava, I can tell from that there's still quite a bit more work to do. In the human gut, in a lot of cases, we'll get 90% plus unclassified viruses. So we can tell they're a unique viral clade, we can phylogenetically place them, but they're so far from anything characterized that they might have family level or worse taxonomy. So there are challenges in isolation and assays. There are challenges computationally. I'm grateful for the NIH for kind of recognizing this and funding things like the HVP, but that's planned as a 10-year, two-phase, 5+5 year project, and there will be plenty of work to do on both angles.
Yeah. So it seems like there's really at all steps in the process some gaps that will hopefully be filled by this kind of project.
I hope so.
All right. Moving on to our next question from Laura. This is, I think, very relevant to kind of like what we think about here at the One Health Microbiome Center with kind of integrating across human, environmental, and agricultural work. But do you expect baklava to work reasonably well for soil viral samples? Since it seems that most viral work is focused on human health, would you share your thoughts on how this impacts environmental research using this tool?
So I definitely expect our coverage to be lower. We've done a variety of both evaluations in Metaflan specifically and Human for normal profiling, functional profiling. We've specifically aimed to improve non-human environments over the past few years. We've increased some coverage, say, of especially non-human hosts from 20, 30% to 60, 80%. We know that tricky environments like soil, we still get, even for bacterial profiling, 10% coverage. And it's again, it's not just us. That is the state of the field at this point. We and I think the field, computational methods in general, at least have a good ability to capture the known unknowns. We can tell how much we're missing. Outside of really deep sequencing to assemble new mags from these environments, though, there's not really any way to capture them. If you don't have a reference and you don't have deep enough sequencing to do assembly, you'll have individual reads that you might not know what they are. And the best you can then do is sort of quantify how much is unknown. So there is really a whole range from North American human guts. We know we get 95% plus coverage at this point, and probably not too far off for that from that for viruses. If you have a very tricky soil sample, that might be 10 or 5% and probably less for viruses.
Got it. Got it. Yeah, thank you for that answer. Um, I'm going to move to a question that I think is kind of interesting, given that we so often in biomedical science use mouse models. Um, so this is from Smita. Thank you for your great insights. Your metagenomic analysis tools are great. Is there any study that you're aware of on mouse gut viromes and any lineage sharing with humans?
Oh, so first part, first of all, thank you for your kind words. I always tell people, tell us when something breaks so we can help fix it. For the first part, yes, there's some proposals in. So there's both at least one or two of the projects in the HVP and a couple of outstanding proposals that I know right now that are specifically for looking at kind of baseline mouse gut viromes. For lineage sharing, no, that was not a thing we had done with mice. I remember seeing many years ago a study on SFB, segmented filamentous bacteria, where it's sort of immunologically unique in a lot of mouse labs. There was a lot of interest in it for T cells. It's individually sufficient for T cell stimulation. There were some studies that looked at lineage sharing of SFB in mice, since it wasn't found in adults. The only two places it was found at the time in many thousands of human metagenomes was infants, so like very early life, and a couple of samples from people who worked in mouse labs. So it is possible, but I haven't seen anything like that for viruses.
Interesting. Okay. Um, yeah, all right. I had a question and it just disappeared. Oh, there we go. This is from David. In your opinion, how could the integration of virome data change our understanding of bacteria virus host interactions? And how can we integrate fungi, and I'm going to extend that to other microorganisms into this picture?
I'm glad somebody brought up fungi. We know they are missed in most settings right now as well. Again, due to sort of some computation, some experimental protocols. Fungi and some viruses are just not really well extracted in a lot of current settings. So it's been a little difficult for us to tell in a bunch of human gut microbiome studies to what degree we're missing them due to computational methods, which I think for developed westernized human guts is not so much the case anymore. But we still don't detect them in many settings due to extraction limitations. So fungi are definitely underrepresented. There have been some great studies of, since they're much more dominant on skin, there have been some great studies there focusing on fungal profiling and starting to chip away at bacterial fungal immune interactions on the skin, where like there and in environmental settings, including things like built environment, those are places where, yes, I think that'll matter a lot, especially bacterial fungal interactions. For incorporating viruses into that mix, we have one collaboration going on now with Seth Rakoff-Newham, who's been very interested in getting absolute quantification. So being able to determine what the absolute rather than relative biomass of those three kingdoms are and how they change over time as a way of starting to get at dynamics and interactions. The spike-in protocol for viruses is unsurprisingly difficult. So we've been working on assay development for that. Check back in a year or two, maybe if we have data there.
All right. This is a question from Florentin about ANPAN. Is ANPAN able to quantify strain richness within metagenomes, or are comparisons limited to only the most abundant strains?
That's again a great question. We tend to focus on the most abundant strain of each species. For a couple of reasons. One, that's a case in which I am a little biased by gut biology. In well-mixed environments like the gut, it is less common for species to have multiple abundant strains. And using skin as an example, that's not the case on skin where there's spatial structure that separates different strains of the same species. So on one hand, it's biologically a little safer to focus on the dominant strain in that setting. The second thing is sequencing depth. It is really tough to have enough coverage to see multiple strains unless you really only have very low diversity or a whole bunch of sequencing. Nicola is working on including multiple strains in we just finished most of the version 4 tools in the biobakery. Multiple strains are on the horizon for version 5, but it's going to be a while because detecting multiple strains automatically and reliably is very difficult. It's very easy to introduce noise from sequencing error that results in inflated strain diversity, and I haven't wanted to go there until we can do it right.
Mhm. As an aside, I am so tickled by the fact that all of your tools are baked goods.
Thank you. Coming up with the names has been historically difficult, but where our job is being replaced with AI, ChatGPT is actually really good at that.
Interesting. Okay. Um, all right. This is from Maria. Thank you for your talk. I was curious to hear your thoughts on the importance of integrating experimental work and validation with metagenomic data. In particular, how do you see this coupling helping to address gaps that purely computational or derivative approaches might overlook or be unable to capture?
Again, excellent question. I didn't show the slide today, but one of the slides I often use to sort of motivate our work has this big funnel where we do work a lot with these large human populations. But my PhD, my background was in Saccharomyces cerevisiae, a very basic model organism molecular biology. So the goal is usually to funnel down from some big population epidemiology to some targets, individual molecular targets that we can take back to the lab. Some studies get there and some don't. Something that I really enjoy working with Wendy is that many of the sort of best experimental setups in mechanistic microbiome studies do rely on notobiotics, controlled communities. I really like some of the work that she and others are able to do now with ASF or modified ASF or OMM13 controlled communities so that you can knock a bug in, knock a bug out, knock a gene in, knock a gene out. One of our lucky success stories recently was able to trace down from the HMP2 population data to a set of 2 gene families in a handful, about a half dozen different bugs that were responsible for a particular type of drug metabolism. And then take those to heterologous expression and cell-free expression, work with Emily Bauskas, who has a nice mass spec assay for chemical modifications, and kind of get the whole way down there. That was in part luck. That's always the goal. Some studies are able to get there and some studies don't.
Got it. Thank you for that answer. Um, we've got about 5 minutes left, so I think we have time for maybe one or two more questions. This one is about baklava again. Baklava is a powerful tool for viral profiling, especially with its combined nucleotide and translated search strategies. But considering the rapidly evolving nature of viral discovery and taxonomy, do you think the current viral reference databases used by baklava are sufficient for accurate and comprehensive profiling? Or do you see some major limitations that still need to be addressed in terms of coverage, annotation, or representation of novel or rare viral taxa?
I like the way the question was phrased because I can answer both. I think there are both settings where there's currently good coverage, not just the human gut, but some of the viral databases that we ingest come from things like the Terra Oceans virome assembly and a set of specifically RNA viral assemblies. So there are certain areas where coverage is already reasonably good. Plus, there's a lot of mileage. We've looked at this a lot for human in difficult environments, like again soil, some marine settings. At the protein level, you can get surprisingly good coverage even when you don't have very close references available. So we know we get pretty good functional profiling even for communities where most of the community is unrecognized. To some degree, that's true for baklava due to the viral and deep learning classification. So there's a glass half full part of the answer where yes, some settings we do pretty well on already. Even in difficult settings, you can kind of run with the fraction of information you get. The glass half empty answer is that yes, of course, we need better viral references. We are set up to continue adding viruses to the database, just like we have with Metaflan over the past few years. That is the kind of thing that, yep, it'll take a while because we're building up from a fairly low level in terms of viral isolates, especially, and the universe of viral diversity is probably higher. So that I do expect to keep being a project for the next few years.
Yeah, yeah, seems like I read somewhere that viruses are the most abundant organism, if you can call them that, on the planet. So I imagine it will take a very long time to characterize all of them.
Rick Bushman, who is one of the founding fathers of viral community analysis, starts most talks by saying exactly that. So
And I think we'll just end on this. Thank you again so much for spending your morning with us, and thank you to all attendees for coming. Um, are there opportunities for collaborating on the Human Virome project? If so, can you talk about some of the areas of need that collaborators or additional researchers could contribute to?
um, that is a difficult question to answer now, given, as Seth alluded to, the sort of uncertainty in the U.S. funding landscape. As of right now, there was so there. If I hop back to the HPP slide and let me know, did that show up correctly still?
Yep, looks good.
Okay. So out of the three arms of the program, the large characterization centers, the functional studies, and the methodological projects, there was a recent second round of RFAs for methodological projects specifically. I believe that just wrapped up recently. The plan has been, if you go to the Common Funds page for this, they have a nice little diagram of how they expect different projects to evolve over the subsequent 9-ish years now, with more tools and protocols towards the beginning, slowly tapering off, more functional studies as we have a better reference base and more information to build on going forward. And then we don't yet have a concrete plan from the Common Fund for exactly what data will be available when, but our mandate is to share all of the data as quickly as possible. So we know we'll be able to start getting data for the project online while a lot of the characterization centers are continuing to operate. So I can't give you a timeline for that other than as soon as possible. For the tools and protocols and functional studies, definitely keep an eye out there. The NIH does have a timeline plan for additional RFAs in both of those tracks.
All right. Well, Seth, do you have any final questions or thoughts?
No, I just want to thank everybody for joining us today. Thank you for all your questions. Know that we're always here, both our centers at Harvard and Penn State, to help the community, and we look forward to joining forces with more of you together. Have a great rest of your World Microbiome Day, everybody. Congrats.